Hello everyone. We have brought for you part 3 of synthetic biology series. The goals of pursuing this field include identification and cataloging important standardized genomic parts for quick synthesis, designing and constructing a simple genome for a natural bacterium, redesigning existing biological parts, expanding the set of native protein functions for new processes, and overproducing natural products by synthetic microbes in huge quanta. After a novel and useful organism is synthesized, we resort to industrial biotechnology for large-scale production of enzymes, polymers, fuels, and growth supplements such as vitamins. Bio-based production of these is much quicker, simpler, cheaper, and eco-friendly than producing the same chemically. That's why industrial biotechnology innovations are now often preferred. However, abiding by the Toxic Substances Control Act is mandatory while doing so. Now, let's discuss the commercial side of synthetic biology. Synthetic DNA manufacturing companies include those selling oligonucleotides, genes, and other biosynthetics such as Blue Heron Biotechnology, DNA 2.0, GeneArt, and Genomatica. Leading consumer companies of such products are those who are building novel biological systems for bioproducts, biofuels, and the healthcare sector, including Amyris Biotechnologies Incorporation, Codexis, Genencore, Life Technologies, Genomatica, Cuteros, Verdezyme, OPX Biotechnologies, Solazyme, and many more. Some biotechnology breakthroughs happened way back in 1970s and saw the emergence of genetic engineering for environmental purposes such as bioremediation. A bacterium degrading many petroleum components was developed, which became the first biotech patent for cleanup of oil spills. In 2003, scientists at the J. Craig Venter Institute, JCVI, led by Drs. Smith, Hutchinson, and Venter, built a fully synthetic 5x174 chromosome in vitro in just a couple of weeks and published this great achievement in the world-famous PNAS, or Proceedings of National Academy of Sciences. In 2004, George M. Church of Harvard and Zhou-Lian Guo of Houston Universities announced that they had invented a new multiplex DNA synthesis technique, which will eventually reduce the cost of DNA synthesis to 20,000 base pairs per dollar. Isn't it amazing? In 2006, J. Kiesling, director of Berkeley Center for Synthetic Biology, constructed a yeast accommodating bacterial and wormwood genes, converting it into a chemical factory to produce a precursor to artemisinin for use as an affordable anti-malarial drug. Then, in 2007, the JCVI published their successful breakthrough in transforming one type of bacteria into another, dictated by the transplanted chromosome, and published their results in the famous journal Science. In 2008, again JCVI created the first synthetic bacterial genome, Mycoplasma genitalium, JCVI 1.0, representing the largest man-made structure, also published in Science. Genome transplantation, synthesis, and assembly are the empowering steps towards the ultimate worthy goal of creating a fully synthetic activated cell. In 2010, scientists at the JCVI announced the synthesis of a single-celled organism based on an existing bacterium that causes mastitis in goats. The genome core was entirely synthetic and was constructed from 3 chemicals in the lab. This organism has got so-called 4 watermarks scripted into its DNA to identify it as synthetic. It took JCVI 15 years to complete this initial project. Much more work needs to be done for mastering synthetic biology, guys. The forthcoming and last part of synthetic biology series would be on social and ethical implications. Hope you find our efforts worthwhile. Please feel free to give your inputs, views, and ask your queries in the comment box. We would love to answer those. We thank you for watching our videos. Till next time, take care and bye.